Today's submodule is the characteristics of the sympathetic nervous system and the parasympathetic nervous system on the cholinergic and adrenergic fibers. All right, folks, let's get started. The receptor's action. When a ligand binds to a receptor, the protein begins to go through conformational change in shape, thereby allowing it to become activated. The binding of a neurotransmitter can open an ion channel in one of two ways. The first way is what we call a ligand-gated channel. This involves the receptor being a channel. When it binds to the signal, it will open up the door, allowing an ion to rush in based on diffusion. The second type is called G protein. G proteins require intracellular proteins to be activated and an effector enzyme to turn on to create a second messenger. Ligand-gated channels can be called ionotropic. For example, binding of acetylcholine will open up both sodium and potassium channels, which allow the passage of sodium into the cell as potassium efflux and exits the cell. The cholinergic receptor is ionotropic, is referred to as a nicotinic acetylcholine receptor. On the right-hand side, you can see here that there's two binding sites for the acetylcholine molecule. There actually are 5 subunits. There's 2 alpha subunits, 1 beta subunit, 1 delta subunit, and 1 gamma subunit, even though the picture does not show you all 5. Sodiums, representing in the blue circle, will go into the cell, while the red circle, potassium, will efflux out of the cell. The other kind of signal transduction pathway utilizes a protein complex called G proteins. They can signal an enzyme cascade, which are metabotropic. These neurotransmitter receptors are separate from the proteins that serve as the ion channel previously discussed. If a cholinergic receptor is considered metabotropic, that means it is called the muscarinic acetylcholine receptor. They can be classified into 3 categories. In all 3 cases, they follow a similar story format. It begins with the 3 subunits, the alpha, gamma, and beta subunits. When the first message, a neurotransmitter or hormone, binds to the receptor, it will activate these 3 subunits. The alpha subunit can then interact with GTP, which causes it to dislodge, and the alpha can bind onto an effector enzyme. In some cases, the beta gamma can be also used as the subunit to affect the effector enzyme. So over here, the beta gamma subunits will be freed from the 3 compartment. It will target and bind onto this ion channel, which can allow the potassium channel to open up, therefore potassium effluxing out. In other cases, the alpha subunit could be the one attaching to the effector enzyme. The 3 categories of G proteins are as follows. The first type is a mechanism called G sub Q protein, which is shown in yellow. This means a stimulation of an effector enzyme called phospholipase C, and the secondary messenger is called inositol 1,4,5 triphosphate, also known as IP3, and DAG, diglycerol, which raises the intracellular calcium level. The second type of G protein mechanism is called G sub I. This is shown in red. Inhibition of the effector enzyme, adenocyclase, will then reduce the amount of the second messenger, cyclic adenosine monophosphate, CAMP for short. This is a derivative of ATP, the energy currency found in our cells. The final kind of mechanism here is called G sub S protein, which requires the stimulation and increasing of the adenocyclase activity, effector enzyme, thereby boosting up the intracellular cyclic AMP availability. All right, let's take a look at each of them more closely. So in this picture, you'll see the G sub I and the G sub S pathway. And the storyline for both G sub I and G sub S are similar. You start off with the first messenger, which could be a hormone or a neurotransmitter. Oftentimes, these water-soluble hormones that are polar-based cannot cross the membrane. And because they can't cross the membrane, they will have to bind to a receptor to begin the signal transduction. The alpha, beta, gamma subunits are shown here. Alpha is interacting with GDP. The moment that the first messenger binds, the GDP is released. Now it allows the alpha to interact with GTP, which will break off from the subunits and target the effector enzyme. This effector enzyme could be an ion channel or an enzyme that will turn on the second messenger pathway. This could result in a change in potential to the membrane and a series of cellular responses downstream, or it can turn on, activate the second messenger, another signal that will turn on, again, a series of signals and transductions downstream in the cell. Let's put the effector enzymes into play. So let's say you have a first message that binds to a receptor. This will activate the alpha, beta, gamma subunit. The alpha will release GDP. It will then bind onto GTP and target the effector enzyme adenyl cyclase. Adenyl cyclase is going to increase the formation of the second messenger, cyclic AMP, from the precursor ATP. In turn, when cyclic AMP is increased, this will allow a series of enzymes called the cyclic AMP-dependent protein kinase, also known as pKa for short, to be activated. And now this activated form of the protein kinase can then phosphorylate other molecules and then produce a cellular response. That is the GS pathway, where you're increasing the availability of CAMP. In the GI pathway, it is all the same, except for now you reduce the availability of CAMP, therefore it will trigger another cellular response. Let's take a look at the GQ pathway. The GQ pathway is also known as a phospholipase C activity. In this case, after the first messenger binds to the receptor, the alpha, beta, gamma subunits begin to activate. The alpha interacts with GTP, will target the effector enzyme phospholipase C. This is a plasma protein which will split the precursor called PIP2. This stands for phosphodidyl inacetyl bisphosphate, which is a component in the plasma membrane of our cells. Once activated, it will create not one, but two second messenger molecules. We have IP3, inacetyl triphosphate, and DAG, diaglycerol. The role of DAG is to go into the cell, and its main function is to, similar to cyclic AMP, activate a series of inactivated enzyme kinases called protein kinase C into the activated form. On the other side, IP3's role is to embed into the cytosol, and it will have an affinity to target the intracellular calcium concentration. It does so by targeting these voltage-gated channels in the endoplasmic reticulum. These pockets store calcium. When IP3 targets these receptors, calcium is going to efflux out of the endoplasmic reticulum and increase calcium ion concentration inside the cell. This will then synergize with the protein kinase C to allow phosphorylation activity downstream in the cell. This will produce some cellular response. Calcium's role is to help activate several forms of the protein kinase C. Cholinergic receptors. There are two types of cholinergic receptors, the nicotinic and the muscarinic. The nicotinic receptors are found on cell bodies and dendrites of all the ganglionic neurons of both the sympathetic and parasympathetic division, as well as on the cells of the adrenal medulla. The first type is called the nicotinic receptors. This is abbreviated as N. There are two classes, N sub M/N sub 1, and N sub N, and also N sub 2. The N sub M stands for the muscular skeletal system, and then the N sub N represents the nervous system or the autonomic ganglia inside the CNS. When a nicotinic receptor binds to acetylcholine, it will produce an excitatory postsynaptic potential in the target cell. All the preganglionic neurons release acetylcholine, which binds to nicotinic receptors on the ganglionic neurons. The second type of cholinergic receptors are called the muscarinic receptors, expressed as the M. Muscarinic receptors are found in all target plasma membranes in the parasympathetic division and a few in the sympathetic target cells. When acetylcholine binds to muscarinic receptors, this induces an indirect response that utilizes the G protein system that we just talked about. It could be the GQ, GI, or GS pathway. There are different types of the muscarinic receptors, and they can either be excitatory or they could be inhibitory. So some will be exciting and some will be inhibiting. All the ganglionic neurons of the parasympathetic division release acetylcholine, which will then bind to muscarinic receptors at their target cells. The only exception to this is the sympathetic ganglion neurons that innervate the sweat glands and some of the skeletal muscle vessels. M1 plays a role inside the CNS and has utilizations for memory development. Its main output is an EPSP, excitatory postsynaptic potential, and it follows the GQ pathway, which means it increases IP3 and calcium levels inside the cell. The M2 pathway is going to be a receptor found strictly inside the heart, and its role is to slow down the heart rate, conduction, and the pumping of blood. It utilizes the GI pathway. It decreases cyclic AMP and drops down calcium. The M3 will stimulate smooth muscle as well as increase secretions within the endocrine and exocrine glands of the body, and this one also follows the GQ pathway. M4 and M5 are strictly found only in the CNS. The M4 utilizes the GI pathway, whereas the M5 uses the GQ pathway. Here is a visual showing you the sympathetic division on top compared to the parasympathetic division on the bottom. You can see that the presynaptic to postsynaptic synapse is a chemical one. It utilizes, again, acetylcholine to nicotinic receptors in both cases, in both divisions. However, in the sympathetic division, the postganglionic neuron can release norepinephrine, and that can target adrenergic receptors in various organs. And the exception to this is only found inside your sweat glands and several small blood vessels inside your skeletal muscle, where it does utilize acetylcholine and muscarinic receptor. So sweat glands and various smaller blood vessels are the exception to this rule. Adrenergic receptors. Adrenergic neurons release norepinephrine, also known as adrenaline, and include all the postganglionic neurons of the sympathetic division, with that exception of the sweat gland. There are two types of adrenergic receptors, which are the alpha and the beta receptors. They can be then further divided into the alpha 1, alpha 2, beta 1, beta 2, and beta 3. All of these are going to utilize G protein cascades. The alpha receptors tend to be a little bit more sensitive to the binding of norepinephrine, which is the neurotransmitter, and the beta receptors tend to be more sensitive to the blood epinephrine, which is going to be the hormone version of that signal. Majority of the epinephrine is generated by the adrenal medulla. Let's take a look and compare epinephrine versus norepinephrine. Norepinephrine is going to be a stronger blood vessel constrictor than epinephrine, physiologically speaking. Norepinephrine will increase the peripheral resistance by constricting down and then boosting up the amount of friction and resistance that the blood has to resist. And it will also increase arterial pressure because your heart will have to work harder to pump the blood. Epinephrine has a slightly stronger metabolic effect. It can help you energize the muscles, increase thermogenic activity, and you can see at a resting state that there is a variation of how much this is made per minute. So on average, you have 0.2 micrograms per kilogram of body weight every minute that is made for epinephrine. And for norepinephrine, you're talking about 0.05 micrograms per kilogram of weight per minute that is generated. Alpha 1 receptors. Alpha 1 receptors generally stimulate smooth muscle contraction. They're found on the skin, skeletal muscle, smooth muscle cells. They're located that cause vasoconstriction. They are also found on the erector pili muscle that result in the pilomotor or the goosebumps that contract when you are scared or cold. They also are found inside the uterus for contraction, ureters, internal urethral sphincter for contracting down and not allowing urination to occur. And of course, they are also found inside your eyes. They are responsible for dilating the pupil and leading into the opening of the iris, what we call mydriasis. Alpha 1 receptors are also found inside the liver, which can spike up glycogenolysis, the process of releasing glycogen and therefore allowing the sugar to be increased in the bloodstream. Alpha 1 receptors follow the GQ pathway. Alpha 2 receptors are located predominantly more of a metabolic function and also found mostly in the CNS, but they're located in the pancreas, which can inhibit insulin secretion, and they're also utilized in the GI sphincters. They could be inhibitory both synaptically and postsynaptically. There are two forms of the alpha 2 receptor. The first form is called an autoreceptor. These are the ones found on the sympathetic postganglionic nerve terminal inside the CNS. What that means is that when you activate the alpha-2 receptor via norepinephrine, the norepinephrine can go back up and bind onto its own self-neuron alpha-2 receptor and cause a negative feedback. This will then create an inhibition effect for that neuron, the presynaptic neuron, to keep on releasing more neurotransmitter. And because of this phenomenon, drugs that are developed to be agonistic that favor alpha-2 receptors could be used to treat hypertension. The other kind of receptors are called heteroreceptors. These are the ones that are found in the parasympathetic postganglionic nerve terminals of the GI tract. They're located throughout the CNS, pancreas, GI tract, and their main role is to inhibit insulin secretion, relaxation of the GI tract, as well as lower the amount of neurotransmitter released via negative feedback. These guys follow the GI pathway. The next set of receptors are the beta receptors. The beta receptors primarily have a stimulatory effect, and they're located inside the heart and kidney, as well as in parts of the salivary gland. Their primary function is to increase the heart rate and force, therefore giving a positive ionotropic and positive chronotropic effect to the heart. That means speeding it up and making it faster and pump more blood. Its role in the kidney is to stimulate the release of an enzyme called renin. Renin takes part in the RAS system, which will convert the liver compound angiotensinogen into angiotensin 1 and then angiotensin 2, which will then lead into the creation of aldosterone. Aldosterone's role is a hormone that will regulate the amount of water reuptake from the kidneys, thereby increasing the blood volume of the body, so it conserves any potential water loss. The beta-1 receptor's function is to follow the GS pathway, so it increases the availability of CAMP. The beta-2 receptors are primarily going to be inhibitory effect and are present inside the smooth muscle plasma membranes of blood vessels and various organs. They're located inside the heart. They're located inside the skeletal muscle, which could lead to skeletal muscle. And the beta-2 receptors are primarily inhibitory in effect, and they're present in smooth muscle plasma membranes of blood vessels, located in the heart, located in the skeletal muscle. They can cause vasodilation, which is a contradiction to the alpha-1 receptor. Alpha-1 receptors will constrict, whereas beta-2 receptors will oftentimes dilate. It will also have a cardio-positive effect, where it increases the acceleration of the heart. It also dilates a smooth muscle inside the lungs, called bronchodilation. It also allows a relaxation of smooth muscle inside the uterus, the detrusor muscles that surrounds the bladder, your GI tract, as well as increasing thermogenesis and lipolysis. It also plays a role to help dilate the lens, allowing you to be able to see far vision. It also plays and synergizes with the alpha receptors to increase glycogenolysis and to release sugars to the body. Beta-2 also follows the GS pathway by increasing cyclic AMP. Beta-3 receptors could be either stimulatory or inhibitory, and they're only found inside adipose tissues, responsible for triglyceride and fat breakdown. Over here is a picture summarizing, again, the sympathetic and parasympathetic division. You can see in the middle, the chemical synapses utilizes acetylcholine. Over here, you have the two classes of adrenergic receptors, the alpha-1, alpha-2, that can bind norepinephrine and epinephrine. And then we also have the beta-1, beta-2, beta-3 receptors that can bind norepinephrine and epinephrine.